anaerobic respiration. We said that once pyruvic acid is made, there are two options. If oxygen is available, then that oxygen will allow that pyruvic acid to be oxidized and then get involved in the citric acid cycle. But if there's not enough oxygen, then we have to undergo what we call the lactic acid fermentation. Here's the problem, is that here's the mitochondria. Normally, NADH that's produced would pretty much immediately hand off its electrons to one of the shuttles. Depending on what cell it's in, it's the FAD shuttle or NAD shuttle, which then that would resupply NAD for glycolysis. But when there's no oxygen available, the NADH can't hand its electrons off to the shuttle because electron transport isn't working. Because there's nothing to accept those electrons, and so everything kind of backs up. So this shuttle stops. And what that leads to then is an increase in the amount of NADH in the cytosol. But eventually what's going to happen is the levels of NAD are going to become very, very low. And what happens if NAD runs out in the cytosol? Let's take a look at this. Maybe this will help. Here's glycolysis. Here's our fructose 1,6-diphosphate. Here's our P-gal, the phosphate. This is 1,3-diphosphoglycerate. Here's pyruvic acid. We know that NAD, of course this is happening over here as well. We know that NAD is reduced to NADH, and P-gal is oxidized to 1,3-diphosphoglycerate. And then ultimately, those two ADPs are phosphorylated to make 2 ATP, right? That's glycolysis. This is the cleavage phase up here. This is the oxidation phase, and this is the dephosphorylation phase. This is just a kind of a close-up of that glycolysis chart we saw. What would happen if NAD ran low or ran out? There would be no way to get to pyruvic acid, which means that there's no way to make ATP. So when we are in anaerobic conditions, there's not enough oxygen to support electron transport. These NADH numbers increase, but the NAD numbers decrease. So in order to maintain a supply of NAD, let me just clean it up here. In order to maintain that supply of NAD, to ensure that there is enough, or that there is still some ATP production occurring, the NADH will hand its electrons off to the pyruvic acid, forming lactic acid. So what happens is NADH is oxidized, pyruvic acid is reduced to lactic acid. That then supplies more NAD for glycolysis. And that can sustain energy production for a short time Typically enough time until your body is able to get the oxygen it needs in the amounts that it needs to maintain full aerobic respiration. And that's something we'll address on Wednesday when we finish this semester up with Chapter 11, looking at how skeletal muscles utilize ATP and where the different mechanisms they use to generate ATP for different types of exercises, different types of activities. Here's what this process basically looks like. If we were to draw out this fermentation reaction, this is what we'd have. Here's pyruvic acid. Each circle, again, represents a carbon. Pyruvic acid would look like this. Oops, should be an OH here. That's pyruvic acid. That's the structural formula. And I haven't given you a whole lot of those with this. What NADH is going to do, it's going to give to the pyruvic acid 2 electrons and 2 protons, basically 2 hydrogen atoms. And what we end up with is this. So that's where those 2 came in, right there. That's the difference between pyruvic acid and lactic acid. Draw it bigger. So there's our carbons. It is in your book. I don't know if they go into the structural formulas. Oops. Carbon, carbon, carbon. Double bond. Remember, each carbon can has 4 bonds. That's pyruvic acid. The 2 electrons and the 2 protons will make 2 hydrogen atoms. And where I've circled is where those 2 hydrogens have been added. That's the difference between pyruvic acid and lactic acid. And I think you have another handout somewhere that shows that other diagram that I had up.
Is there a reason why the hydrogen doesn't bond to the oxygen or whatever on the inside?
It's been a while since I've had my organic chemistry, but yes, there is a reason.
Okay.
And there's a reason that it goes here. And organic chemistry would explain to you why it goes there. And I don't remember. It's been a while since I've had organic chemistry, almost 20 years. I'd like to take it again, actually. So this process is important because it replenishes the NAD necessary to keep glycolysis going, which is pretty cool. But here's the issue, is that the lactic acid can build up in the cell, and as the cell gets more acidic, it affects the enzymes that play a role in all these processes. So the cell has to get rid of that lactic acid, and so it diffuses from the blood, or from the cell to the blood, and then to the liver. And then the liver actually converts it back to pyruvic acid. It kind of looks like this. Let me jump ahead. Looks like this. So here's glycolysis. Here's our energy that's produced. Here's the NADH. Here's the replenishment of the NAD, which goes back up and is used up here. Here's the lactic acid, diffuses, goes to the liver. Eventually, it's converted back to pyruvic acid. Now, we'll come back and talk about this slide in a little bit, the rest of it, but that's basically what happens. So there's some checks and balances. The problem is that eventually lactic acid does build up in the blood, and that can actually affect endurance. And we'll talk a little bit about that on Wednesday as well. It's called the lactic acid threshold. Okay, so the solution to not having oxygen is the lactic acid fermentation, and the solution for the buildup of the lactic acid and the effect change in pH is to shuttle that lactic acid out of the cell to the liver, where the liver is then going to process it and convert it back to pyruvic acid, where eventually it can enter into the citric acid cycle or into the matrix reactions, or potentially even be converted to glucose. Now, some cells are actually able to use lactic acid. The heart can actually use a little bit of lactic acid to generate ATP, mainly during strenuous exercise, and during a heart attack as well. Some lactic acid would be, lots of lactic acid would be produced, but eventually that's not enough to sustain the heart either. But some cells are better adapted to levels of lactic acid in them than others. The skeletal muscles have the best tolerance to lactic acid. As you exercise and get in shape and increase your endurance, your tolerance for lactic acid in your skeletal muscles increases. Cardiac muscle can survive longer than nervous tissue. Nervous tissue is what we call an obligatory user of glucose. It specifically prefers to use the glucose to generate its ATP. So that can survive. That has a very, very low tolerance, very low threshold for lactic acid. Now, there are some other organisms that have similar pathways, and this is where the term fermentation comes from. Because in some organisms like yeast and bacteria, they undergo a different type of reaction. It's very similar. It's for the same reason. What happens is that, again, in anaerobic conditions, the yeast will produce pyruvic acid, but in order to maintain the levels of NAD, they have to somehow do something with those NADHs. And because it's an anaerobic condition, they can't undergo the electron transport process. So, just like we have with lactic acid fermentation, what the yeast do is they actually cleave off a carbon molecule, actually cleave off carbon dioxide, and actually donate then the protons and electrons to this molecule here, acetaldehyde, to produce ethanol. So I make beer. I've talked about my beer making a few times in class. One of the things that you do when you make beer, or what you see, is that when you add your yeast, the yeast are consuming the sugars in the wort, which is the solution that's not beer yet. So they're using the sugars, but because they don't have oxygen, they actually use it up. Because they don't have oxygen, they have to undergo this fermentation pathway. And so the carbon dioxide you get when you ferment and produce beer or wine comes from this reaction. And then the reason we get the ethanol in the beer and the wine, the alcohol in the beer and the wine, is because the NADH reduces this acetaldehyde to ethanol. And then we oxidize NADH back to NAD. So back in the day when water quality was very poor, hundreds, thousands of years ago, they used these fermentation reaction because the alcohol and the yeast would actually clean up the water. They'd boil the water as part of the process. But the ethanol would actually kind of preserve the beer and the wine, and they would actually have a healthy drink to drink. It'd actually prevent them from getting sick from drinking bad water. Okay? Anyway, that's not going to be on the test. I just like talking about that, just because I like talking about making beer and wine. I'd like to actually offer a class, maybe a continuing education class on beer making here. Yeah. Well, there's a ton of chemistry in making beer. There's a ton of chemistry, not just organic chemistry, but there's acid-base chemistry and ion chemistry. There's all kinds of stuff looking at water quality, and you can get a whole degree, I think a master's degree in brewing science at UC Davis in California. I've actually looked into it. The only way I'd be able to go is if I got divorced, so that's not going to happen. My wife won't let me spend a semester or a year out in California. Can't. You got to be there. You got to be there. Anyhow, trying to get the college to pay for it for me. Trying to get them to start a program here, like a brewing certificate. Apparently, there's a couple breweries going to open up in Muskegon. I heard that. So, yes and no. When you ferment, question was, does the carbon dioxide stay in the solution? When you ferment, no. I put a little airlock on, and so I can actually see there's liquid in it, and I can see the bubbles. But when you carbonate the beer, the reason it gets foamy is you can force carbon dioxide into it with a tank, or you can add an additional little bit of sugar solution once it's in a closed container, and then because it's capped off, carbon dioxide builds up, and then it just kind of, as it keeps building up, it has nowhere to go, so it goes into solution. So
you're sure that's not going to be
maybe extra credit. I'm positive that won't be on the exam. But that's where this idea of fermentation came from. Calling it lactic acid fermentation. It's the same basic process, except we don't produce alcohol, we produce lactic acid.